Hello everyone, my name is Meetum, and I'm gonna just do my biochemical test by just drawing on this paper. So I'm gonna pretend that my pen is gonna be my inoculating loop or needle, depending on which situation I'm gonna use it for. So in this case, I will use it as an inoculating loop. So before I do anything, I'm gonna put my loop into the incinerator for like 10 seconds, and then just put it to the side, and then I'm gonna uncap my source culture broth and then put it to the incinerator with the like cap open, or the glass towards the incinerator, and take it off from, and then and start inoculating by just I'm gonna draw my needle. I'm gonna go whoosh 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 like that to get my sample in, and then put the tube back to. And once I take out the inoculating needle with one hand, I'm gonna put the source tube back to the incinerator and then close the cap. I'm gonna start inoculating with my Stark agar plate. I'm just gonna do a general, streak like this, because I don't think I need to do quadra streak or T streak. I'm just gonna wait for results to come out. And after I incubate this for 24 hours, I'm gonna wait for 24 hours. I'm gonna add several drops to grams iodine to it. So I'm gonna go right here, and it's gonna be right there. I'm gonna add and try to and see the results after it's been incubated. Well, before I incubate it, after I'm done inoculating, I'm gonna put my inoculating loop through the incinerator, and then I'm done for this part of the biochemical test. So moving on, I'm gonna do the same steps that I did before of putting the inoculating loop through the micro incinerator, and then capping and uncapping with the near, and disinfecting the top with the cap off, and just putting it towards the micro incinerator, and doing the same streak that I did before, so I can get the culture into the loop. And then all I'm gonna do for the casing in this case with my newly inoculated loop is I'm just gonna do that, and then sterilize the loop after I'm done doing that for the casing and agar test. So that's what I'm gonna do, and this doesn't need any reagent. It's gonna go into the incubator. And then moving on to the tryptophan broth, I'm gonna make sure I micro incinerate my inoculating loop, and then before I touch anything, open the cap, putting it towards the micro incinerator, and then after I'm done, sterilizing, I'm gonna put my loop like that little shake around like I did before, putting on an inoculating loop, and then I'm gonna insert my loop here and just kind of swish it around so I could broth in the switch. I'm gonna put my inoculating loop here and then switch it around so I can get the culture into the broth. And then after that's inoculated, before I inoculate it, I'm gonna have to put the cap towards the incinerator and close the cap. Put the cap towards the incinerator, and then not the cap. It's gonna melt off. Put the tube near the incinerator before I can inoculate it, and then so after I'm done inoculating, I'm gonna put the test tube near the incinerator and then close the cap, and then incubate this for 24 hours. And after the 24 hours are done, I'm gonna add this Kovac reagent after it's been incubated and been sitting around, and then before I incubate it, I'm gonna disinfect my micro incinerator group. So that's how I'm gonna do it. Moving on to the next test, to the urea slant. This time, my pen would be an inoculating needle. So I'm going to put my inoculating needle towards the micro incinerator, open the cap, and then have the source culture, and I'm just going to maybe just go in there, take a little bit of sample of what I need. I don't need too much. I don't need to take out too much sample, and that will just be too much to lose. And then I'm gonna proceed to inoculate the needle here, right there to the urea test tubes, and then I'm just gonna open the cap, put the cap towards the micro incinerator, and then I'm just gonna do one slide up right there, and then from the face view, it's gonna look like I did this when I inoculated it. And I'm gonna make sure I go 3/4 of the way down, so I can make sure the urea can really, like, go through its metabolism and stuff like that. And yeah, and then after I'm done inoculating that, I'm gonna put my needle through the micro incinerator. So moving on to the catalase. In this case, I would not use my ink inoculating needle because I can just use a wooden stick. So what I'm going to do is I'm just going to do the same procedure of just touching the wooden stick to get some sample on it. And then once that one end of that wooden stick has the this is an immediate test where I can just immediately tell rather than waiting on incubation, unlike the urea slant. So I'm gonna put a drop of hydrogen peroxide on top of the stick and just go drop that. I'm just gonna pretend these are droplets, and I'm gonna wait for oxygen to form or bubbles to form. So if I see bubbles to form, that means oxygen is present in this bacteria. So that's done. I'm not gonna try to burn off the stick. I'm just gonna throw away the stick. So for this one, I'm just gonna get the cotton swab and then put it into the source tube and just touch it slightly just to get enough culture into the cotton swab for the oxidase reaction. So I'm gonna once I got this culture on top of the cotton swab, I'm gonna put the oxidase reagent on top and wait for a color change to happen, so I can see if the electron transfer chain occurred for this bacteria that's on the source. If this is an immediate reaction, so I don't have to put it through an incubator, and I could just wait for the result to happen, like a blue or dark purple color change. And so yeah, and then I'm just gonna dispose of that to the trash can. So I'm gonna have, so the next part I'm gonna do is the Durham sugar tube test, where I'm gonna use an inoculating loop to just to get, for all of these procedures, I'm gonna use an inoculating loop. So I'm gonna put my inoculating loop through the micro incinerator, and then open the cap and go through the procedure of closing the cap and sterilizing the tube. And then I'm gonna since it's a slant, I'm gonna carefully go in and just take a little bit of sample, and then put it into here, and then just inoculate it and just swish it around like I would usually do with the inoculating loop, so I can get cultures spread out in the test tube as much as possible. And before I open this cap, I would just do the same procedure of opening the cap towards the micro incinerator and closing it. And then this test tube would be incubated so you can wait to see the results. So that's what's gonna happen to that one. So now I could move on to the VP test, the Volksboxer test. Yeah, I'm sorry, I don't know how to pronounce that. Sorry. So what I'm gonna do is I'm just gonna do the same procedure, just sterilizing my loop, opening the cap procedures towards the micro incinerator, and then taking a small sample of the source culture, and then inoculating it into here, swish it around with the inoculating loop. And then I'm gonna have to incubate this for 24 hours, And then after that incubation period is done, I'm gonna add this 10 drops of this reagent to here, and after that, I'm gonna add 10 drops of this potassium, like KOH, into here. Here. So this is VPA. I'm gonna add this VPA reagent first, 10 drops of that, and then this KOH reagent, 10 drops of that into this Vox phosphor test, and wait for the results to see a color change, or just to see like any observable color change for that test there. For the methyl red test, I'm gonna go through the same procedure of inoculating, like first sterilizing my loop before I can do anything, putting my source slant in towards the micro incinerator, and then taking out my inoculating loop to get a little bit of source culture, and then I'm gonna put it towards here, and then I'm gonna switch it around. Make sure before I insert any culture into this, I'm gonna sterilize my tube with the cap off with the micro incinerator. Insert that, and make sure this test tube gets incubated for at least 24 hours before I put any reagent. After that incubation period is done, I'm gonna add 20 drops of methyl red and wait for an observable change for about 30 minutes. So yeah, that concludes this part of the biochemical test. Now I'm gonna, for the nitrate reduction test, I'm gonna use an inoculating loop here. Use one of these. So I'm gonna put that through the micro incinerator. Make sure it gets sterilized. Open up my culture source, like where I get my culture. Put it into the micro incinerator and cap. And then now I would put that loop just enough to touch the. I wouldn't try to dig in to grab any culture. I'll just like try to touch it and get some into the loop as possible, like not much as possible, just enough to like have some culture into the loop. I'm gonna put that loop in here, swish it around before I insert my specimen into the nitrate broth. I'm gonna do the capping procedure towards the micro incinerator, and then I'll inoculate it and just do a swishy thing inside the broth. And after I inoculate it, I'm gonna wait for 24 hours for that nitrate broth to be inoculated. And after that wait is done, I'm gonna add sulfuronic acid, 20 drops of that into here. And then after that, I'm gonna add another 20 drops of alpha thilamine reagent into here, or nitrate A reagent first, and then nitrate B reagent second. Both 20 drops. And if there is not a color change after those 40 drops of those both reagents, then I would then add zinc dust into the broth. But that's only I only add zinc if there wasn't a positive positive reductase result. So yeah. So for the next part, I will repeat my inoculating loop sterilize procedure and I'm gonna test tube sterilize procedure with the micro incinerator, and then inoculate this just enough for it to get a little bit of culture in it. And then I'm gonna go. I'm gonna use an inoculating like a needle, actually switch it and sterilize my inoculating needle, and just to stick this in and just get enough. And then I'm gonna go stab it like that, and then maybe do a little swish when I come back up for that citrate test, and then I'm gonna put that to the incubators for it to be done. So for any test tube I'm gonna open, I'm just gonna make sure it's sterilized at the top. Before I inoculate it. And yeah, I'm just gonna stab it and start streaking inside the test tube. Okay. Moving on, I'm gonna do the motility test. So for the motility test, I am gonna use an inoculating loop. That's my inoculating loop, right there. And then just do, before I do anything, sterilize my loop. Sterilize my inoculating needle, and open up my source culture, and then sterilize the cap area. Like, take off the cap and sterilize the test tube opening area. Those procedures are always going to be repeated. And I'm gonna just stick the needle in, get my culture into the needle, and after that, I'm only gonna make, I'm gonna do this cap sterilizing test tube opening procedure, and then I'm gonna stick my needle in. But I'm not gonna do the going back and forth streaking. I'm only gonna do the one line of sticking it into the motility test tube, and then making sure it's the same line that I draw it out. So once I draw out my needle, I'm gonna make sure it's the same line. And same thing with the TSI slant. Once I sterilize my needle and make sure that the top opening of the test tubes are sterilized as well, I'm gonna go in with my inoculating needle, just go enough just to get some culture on it. And I'm gonna do the same thing that I did for the the same inoculating procedure, while I just make sure that there's only one line that I streak up. So from the side view it looks like this, but the face view, I'm just gonna go make sure it's a straight line. Like, I come back up the same streak line. So yeah, and after I'm done, I'm gonna put my inoculating needle into the micro incinerator, and then do the opening cap sterilization procedure for micro incinerator. So I would not cross-contaminate these tests with the culture. Once I take it out, I'm gonna make sure before I do that, sterilize my needle, and then proceed to this procedure. So this concludes my biochemical tests for microbiology. So thank you so much for watching.